All right, gentlemen, this is the second of two videos on section 17.1 in your textbook, and for gene expression. And this half of the two videos is going to talk about the processes of transcription and translation. Now, transcription, we'll start with transcription. It occurs in the nucleus, and essentially what it is taking the DNA code and creating an RNA transcript. All right, that's why we call it transcription. All right, so going from DNA to RNA. And the reason that we do this is because RNA is kind of the midpoint or the bridge between the DNA on one side and the protein on the other. All right, so it's the bridge between the genes and the proteins for which they code. Now, the process of transcription produces a particular kind of RNA, which is called mRNA or messenger RNA. Now, once the messenger RNA has been created and processed, it then leaves the nucleus and goes to the ribosomes. Now, recall from the fall that we talked about the purpose of a ribosome is for protein synthesis. Right? Ribosomes are really just little protein factories. And so that is where translation takes place. And translation is the synthesis of a new polypeptide. Now, this process varies in prokaryotes versus eukaryotes. In a bacterium, the process of translation can actually begin even before transcription has finished. But in a eukaryotic cell, because there's a nuclear membrane that separates the DNA from the ribosomes, the process occurs in two distinct phases. Now, before the RNA can actually get through and go to the ribosomes, it does have to be processed, and we'll talk about that as well. So the next couple of slides just kind of illustrate what goes on with this. We start out with the DNA molecule, and you can see that located here. And in the process of transcription, this takes place and we produce mRNA, and then eventually translation, and this is all done inside the cytoplasm of the cell. This is for a bacterial cell. And so all of this can occur actually simultaneously. Now, in a eukaryotic cell, you can see that the nuclear membrane, which is located here, separates the DNA from the cytoplasm, which is where you would find the ribosomes. And so in the first phase, the DNA is transcribed, and we produce a molecule we call the pre-RNA, or mRNA. We then process the RNA by removing little bits and pieces. Notice very carefully, this bit and this bit have been excised. They've been cut out during this processing part. The mRNA then diffuses through the nuclear membrane and reaches a ribosome, where it can be translated into a baby protein, a polypeptide that you see here. So the original piece of mRNA that's produced is called a primary transcript. All right, and so that's what you would see here. This is the primary transcript. Once the primary transcript has been processed, it then will move through the nuclear membrane and find a ribosome to produce the protein. But always we go in this order, from DNA to RNA to protein. So how does this actually work? All right, how do we get the instructions for building a particular protein with its very distinct sequence of amino acids into a protein? Well, we know that there are 20 amino acids, but only 4 nucleotides in a DNA molecule. And so how do we figure out how many nucleotides are needed to code for a specific amino acid? Well, it turns out that the answer there is 3. Okay, you need 3 nucleotides to code for a given amino acid. And we call these little triplets, and I love the fact that they are triplets. We call these triplets of nucleotides codons. All right. So the flow of information from a gene to a protein is based on what we call a triplet code. It's a series of non-overlapping 3-nucleotide words. All right. Think about this. I always illustrate, to my students. I have 3 letters. And yet, if I organize these 3 letters in 3 different ways, I can produce 3 different words. All right. And so it's not that an adenine, and a guanine and a cytosine together always produce the same amino acid, because you can arrange them in different orders and code for different amino acids. And so the words of a gene are transcribed into complementary, non-overlapping 3-nucleotide words of mRNA. These words are then translated into an amino acid, and many words form a chain of amino acids, which makes the polypeptide. Just to illustrate that here, we start out with the DNA here, and you can see that there are several genes located here, and we're going to concentrate on the 3rd gene, which is located here. And so we have the DNA template strand. We break this in half, essentially. We cut it right down the middle, and then we build an mRNA that's complementary to the top strand of the RNA. Notice that where A goes with T, in the mRNA we get uracil. C goes with G, but there's still a G. C goes with G, there's still a G. A, T, U, and so on. G goes with C, it's still a C, and so on. But it turns out that UGG codes for tryptophan, and UUU codes for phenylalanine, and GGC codes for glycine, and UCA codes for serine. Okay, and so the creation of the mRNA code is transcription, and then the creation of the protein from the mRNA is called translation. So there are 2 strands of DNA, obviously, and we ignored one of them. The one that is being used for the ordering of the nucleotides is called the template strand, okay? And that's the one that is responsible for production of the mRNA transcript, all right? In this case, it would be the one that I've drawn the arrow next to, the top one. The template strand is always the same strand for a given gene, and this strand is used as the template, and it's determined by the orientation of the enzymes that transcribe the genes. All right, and this always goes from 5' to 3'. All right, look at it again, 5' to 3'. Now, the non-template strand is called the coding strand because the nucleotides in this strand are the same as the ones that you find in the mRNA. The only difference is that wherever there's a thymine in the DNA molecule, there's going to be a uracil in the RNA. And so you then have each codon specifying 1 amino acid. Now, if you've done the math quickly in your head, you realize that with 4 different RNA nucleotides, you can arrange a grand total of 64 different codons. But there aren't 64 different human amino acids. And so there are replicates, right? There are some codons, or there are multiple codons, I should say, that code for given amino acids. And this is the coding table, all right? All 64 codons were deciphered in the early 1960s. 61 of them are codes for amino acids, and there are 3 that are stop signals, and 1 which always codes for methionine, which is the start signal. All right. The genetic code is redundant, All right, as I said a minute ago, more than one codon can code for a given amino acid, but no codon codes for more than one amino acid. All right, and the codons, of course, must be in the correct reading frame in order for a given amino acid, a given polypeptide to be produced. But notice we've got 2 that produce phenylalanine here, 2 for leucine, another 4 for leucine. So there's 6 for leucine, 3 for isoleucine, 4 for valine, and so on. You can see, and all 20 amino acids are represented in this table of 64 codons. All right, and again, the only 3 codons that don't code for an amino acid are the 3 stop codons: UAA, UAG, and UGA. Now, this genetic code of all these 64 codons is universal. All right, and it's shared by all living organisms, from the simplest of bacteria to the most complex of animals. And interestingly enough, you can take genes from one organism and actually insert them into another. There are 2 examples in the picture on the right. There's a tobacco plant that's expressing the firefly gene that makes the butt of a firefly glow in the dark. And this pig's nose and hooves are expressing a jellyfish gene, which also allows it to fluoresce. All right, and so the genetic code is universal, and that's something that you definitely need to keep in mind. All right, my friends, that concludes this particular video. I hope you've enjoyed it, and I will see you sometime soon.